Tumor suppressor genes. Okay, first and foremost, microcell biology, what have we got? Tumor suppressor genes are recessive. Why is this important? Because we need to have both genes to therefore be expressed. This is important, tumor suppressor genes, because of something called Knudson's two-hit hypothesis, and that was based on the concept of the retinoblastoma, which is a rare tumor occurring in children, typically under 5 years of age, and this is a cancer of the eye. Detected usually, and it was suspected when children would have a photograph taken and the red reflex would then be absent. So tumor suppressor genes are recessive, which means we need a homozygous recessive for it then to be expressed. Knudson's two-hit hypothesis. Okay, the two tumor suppressor genes that we've got is pRB and so P retinoblastoma gene and the P53. Now, just before I go there, I'm just going to elaborate a little bit on the whole tumor suppressor gene business itself. So tumor suppressor genes, it is exactly as it says on the tin. These guys suppress the formation of a tumor. Remember, a tumor is a mass of cells which have developed due to uncontrolled mitosis. In contrast to tumor suppressor genes, so if these guys are going to suppress the formation of a tumor, which is again uncontrolled mitosis, these guys must be the brakes. Okay, these guys act as though the brakes. These are the brakes of the system of the cell cycle. This is in contrast to your proto-oncogenes, which become oncogenes by a mutation. And again, an oncogene is a cancer-causing agent, like an oncogen. And if these guys are the brakes, those guys must be the accelerators. Okay, so those guys accelerate mitosis, and in the event that they become mutated, proto-oncogenes become mutated to oncogenes, they then accelerate mitosis. Back to tumor suppressor genes, this becomes all important. So let's start with pRB, the P retinoblastoma gene. So this guy actually sits inside the cell, not on his own, but rather as a complex with pRB E2F. What is E2F? E2F essentially is a transcriptional factor. CDK4 comes along and it breaks this bond, okay? So E2F is a transcriptional factor. And if it's a transcriptional factor, what is it going to do? It's going to promote transcription. When do we do transcription? Well, transcription is the first part of protein synthesis, if it went for a better term, with the translation following, or otherwise, under normal circumstances, E2F is going to bind to this DNA molecule and then actually promote the cell cycle replication. So what happens under normal circumstances? It sits there doing nothing, okay? CDK4 comes along, it breaks this little connection between these two guys, splitting it into pRB and E2F. E2F is now free to go and attach to the DNA molecule and therefore allow the transcription to occur. And this will then take the phase from your G1 phase into S phase, i.e. synthesis phase. Remember, we had the G1 phase, the S phase, G2 phase, and the M phase. G1 phase where the cell grows, G2 phase generally where the DNA synthesizes, S phase where the DNA synthesizes, G2 phase then essentially where the cell grows and the checks are done and the organelles continue to grow, and M phase mitosis, i.e. prophase, metaphase, anaphase, and telophase followed by cytokinesis. Now, if you look at this, if this is going to go from G1 phase, okay, to S phase, this guy must act over here. And if you remember from last year, G1S was a checkpoint. So the checkpoint controlled here, G1S checkpoint, is actually controlled by the pRB. And that's that. P53. P53 actually is the guardian of the cell. This guy, what he does or she does is they actually, once you've actually formed the DNA has synthesized, it needs checking, the quality assurance. If the DNA is defective, what's going to happen? Well, this guy is going to say, okay, there's a defect, let's repair. Okay, let's repair the DNA. If, however, the defects are significant, it's going to say, look, this is absolutely useless, let's scrap it. And if you're going to scrap it, it's going to stimulate the process of apoptosis. Well, again, because this guy's a guardian of the angel, not going to do the work himself or herself, instead it's going to allocate or delegate that work to this guy called BH3. What does BH3 do? Well, he goes and interferes with these anti-apoptotic proteins. So under normal circumstances, You've got the cell there, and it's not going to undergo apoptosis, so your anti-apoptotic proteins are going to be in action. However, BH3 says, let's knock you guys out and let the process of apoptosis happen, because the DNA is a complete mess, made a pig's ear out of it. Okay, that's one of the things that can happen. The other thing that can happen is, like we said, repair. Now, if I'm going to undergo repair, then I need to enter cell cycle arrest. I need to put the cell cycle on hold, and I should carry out the repair for it then to be checked again. So cell cycle arrest, we can enter two phases. We can enter the phase of senescence, or we can enter the phase of quiescence. So what's the difference between senescence and quiescence? Well, senescence essentially is irreversible. It is permanent. The cell will never come back. Okay, that's it. That's the end. It will never differentiate into anything. Quiescence, on the other hand, is actually in fact reversible. And in quiescence, there's another kind of within that quiescence state, you can have a state of dormancy. Dormancy essentially is the cell goes into a sleep mode. The difference between now, essentially, this whole thing is reversible. As I say, quiescence is reversible. But the difference then between dormancy and quiescence is such that dormancy, because it's asleep, it needs waking up. It needs triggering. Come on, wake up, wakey, wakey, wakey. So therefore, it requires a trigger. Whereas quiescence can come back from the cell cycle rest into cell cycle replication without the trigger. And that's your P53. So there you have it. Tumor suppressor genes in a nutshell. pRB, P53, what they do. Oh, of course, P53, as you've already now hopefully gathered, and this acts, let's do that, this acts here, the second checkpoint, the G2M checkpoint. Why? Well, of course, I've got the DNA, it's been synthesized here, and now G2 to M is going to, in that mode, before it enters the actual process of mitosis, and the two cells that are going to divide are going to have this DNA, so I need to make sure it's all good quality DNA and non-dysfunctional. I hope that makes sense, and thank you for watching.